Hello everyone, welcome to tonight's Science News Special. I'm your host, Zhong Yizhang.
I'm Ruo Qinglan.
In the wake of the COVID-19 epidemic, humans' battle against viruses never stop. Tonight, we will explore a technological breakthrough that could change the future: broad-spectrum virucidal materials. Later, we will connect with Dr. Lauren Batts, an expert in the field of spectrum virucidal materials research, for an in-depth look at the past life of this material and its future potential. Although it has been more than 2 years since the COVID-19 virus epidemic, I believe most people in front of TV still remember the days of home isolation. The COVID-19 epidemic highlighted the urgent need for low-cost tactics to combat viruses, as it proved that not every family in the world can afford the cost of vaccines. And broad-spectrum virucidal materials can provide as a solution to fighting viruses. Broad-spectrum virucidal materials are engineered substances capable of inactivating or suppressing multiple viruses. This material serves as a cornerstone in both preventing infections and reducing viral spread, protecting not only public health but also critical bases in healthcare and daily life. Next, we look at the development of broad-spectrum virucidal materials. The journey of broad-spectrum virucidal materials has been remarkable. Initially, the focus was on external disinfectants, like alcohol and chlorine-based solutions like bleaches, used to sanitize surfaces. These materials effectively reduce the spread of pathogens on objects, but had limitations in protecting the human body directly. With advancements in science, the focus shifted towards internal use agents, such as broad-spectrum antiviral drugs and coatings that are biocompatible and safe for humans. One of them is broad-spectrum virucidal materials.
To understand how broad-spectrum virucidal material works, let's first explore the process of virus invading host cell. The virus attacks to the specific receptor on the host cell using its spike protein, like a key fitting into a lock. Once attached, it enters the cell and releasing its genetic material. Inside the host, the virus hijacks cell's machinery to produce viral protein and replicates its genome. Newly formed viruses are then released, often destroying the host cell and go on to infect neighboring cells. Broad-spectrum material stops this cycle at the very first stage. This material will block the virus' ability to bind to the host cell. However, this approach is reversible, meaning it stops the virus from entering but not directly destroying it. Current effort is focusing on modifying this material to make them can directly kill the virus. Now, let's compare this material with current using treatment, vaccine, and drugs. Broad-spectrum antiviral material is better than them when it comes to timeliness, target virus range, resistance to mutations, and drug resistance. This is because they work on the basic structure of virus instead of the specific antigens. This making them a great option for fighting virus at an early stage of a pandemic. However, the details about how they kill virus are still unclear. Today, it's our honor to invite Dr. Lauren, a PhD student from the University of Manchester's Department of Material, to talk about her research on broad-spectrum antivirus material. Let's take a look at the interview video.
Yeah, it is. So it's based on, most viruses, to attach onto a cell, what they will do, so you have your virus, and your virus sort of has little spikes on the outside of it. That will attach to receptors on the host cell. However, to help the virus make contact with the cell, there's these things called proteins on the cell surface, and they're called heparan sulfate proteoglycans. But I say HSPG because it's a mouthful. However, these have these things called heparan sulfate on them. So it's a polymer, it's a polysaccharide, so it's a carbohydrate that's really highly negatively charged. Most viruses use this sort of heparan sulfate mediated entry route to get into the cell. Through what we're trying to do is essentially use that by having polymers that are highly negatively charged to simulate that. So that's the reason why like heparin is normally used, because heparan sulfate and heparin are functionally pretty much identical. Like, yes, there's slight differences, but they're functionally identical. So that's the reason why that's normally used first. But again, going back to that virustatic mode of action. Yeah, so the mechanism that we're trying to use is very, very highly conserved with viruses. Like, the likelihood of a virus gaining resistance to something that is so unique and actually quite non-specific to a virus entry and has been that way for as far as we can remember is very low. Like, it's so the issue with things that are like our current options is that they're normally super specific to like a virus glycoprotein that's like on the surface of the virus, and it's specific to that one specific protein. And that's the reason why the virus can like adapt super quickly and change up its genetic material to change that protein.
Broad-spectrum virucidal materials hold great promise as a biocompatible, human-safe, yet effective material to combat viruses. Not only limited to traditional aspects, such as air purification and medical materials, it can also be used in other virus-prone environments, such as food protection to prevent food poisoning and provide cheap, clean food and water to developing countries.
Research into broad-spectrum antivirus material not only provides us a new weapon against future viral challenges, but also show us a great potential of science and technology to improve human life. Many thanks to Dr. Lauren for a wonderful exchange, and thank you to all viewers for tuning in. We will see you next time.